Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Roy Sevilla from Avid Bioservices.
Thank you, Leah, for the kind introduction, and thank you everyone for joining today's webinar. As Leah mentioned, my name is Roy Sevilla. I am a Senior Manager at Process Development, focused on upstream sciences involving technology transfer, process development, and scale-up. It's my pleasure to speak on behalf of Avid Bioservices to highlight our technology transfer perspective and high-level procedures. As a contract manufacturing and development organization, we put a lot of focus on technology transfer and see it as one of our core competences. Successful technology transfer is essential to enable rapid manufacturing of biopharmaceuticals for early and late-stage clinical supply. When customers approaches Avid Bioservices to transfer an existing process, we approach the technology transfer as a structured way involving a cross-functional team from all key functions of the company. This helps ensure the ability to understand and execute against project requirements, mitigate risks, deliver the project on time, and substantially increases the probability of getting it right the first time. So Avid has created a number of tools and best practices that enable the team to plan and deliver successful technology transfer and ultimately secure clinical supply for our clients in a timely fashion. As a quick introduction to Avid Bioservices, as I mentioned, we are a dedicated contract development and manufacturing organization focused on development and cGMP manufacturing of biopharmaceuticals, products derived from mammalian cell culture. Our company provides a comprehensive range of process development, high-quality cGMP clinical and manufacturing services for biotechnology and biopharmaceutical industries. We are located in Tustin, California, which is in Orange County. We are strategically located just 5 miles away from John Wayne Airport. We see this as a logistical advantage as our clients can easily travel for face-to-face meetings held during critical project milestone discussions. We're about 200 employees, and we have 2 operational commercial manufacturing facilities, plenty of capacity, and we have exceptional quality systems and exceptional history of regulatory inspection. We have a variety of stainless steel capacity from 100, 300, and 1,000 liters, and a single-use bioreactor options from 200, 1,000, and 2,000 liter sizes. We see ourselves as well positioned to meet the growing market demands given our investment in single-use and small-scale bioreactors. So we see these key ingredients listed here on this slide as elements for a successful technology transfer. It's important that communication is well established between the process sponsor or donor to our facility. Initially, a gap risk assessment is performed where, in the real world, in most cases, technology transfer programs developed or manufactured from other facilities is not a plug-and-play process, therefore requires a a gap assessment or facility fit assessment. Batch records are shared during this technology transfer process, where Upstream executed batch records, harvest clarification and purification, where it states process operating ranges and testing specifications, bill of materials, where it lists a complete list of cell culture media and buffer feed components, equipment data from the process transfer site, where we can perform equipment gap and trend analysis, buffer media recipes, including formulation records and cell culture media and feed components, and process parameters, where we can identify scale-independent process parameters such as pH, bioreactor temperature, dissolved oxygen, and scale-dependent process parameters such as feed volumes, mixing, and gassing and aeration. In this process, in this technology transfer program, it's also imperative that we receive some process data that helps support process actions, triggers, specifications, and so on and so forth. And that leads to then some process description and development reports to help guide our risk assessment and gap analysis. And during the tech transfer program, we have cross-functional teams from key areas of the organization to help facilitate the technology transfer. And it truly is a hand-in-hand team effort where good and continuous communication between the key functional areas is imperative. Here at Avid Bioservices, we have a process development team which focus on the transfer and scale down production models and gather scale-independent process information. We work with manufacturing quality sciences that verifies that the process is operating as designed and gather scale-dependent process information. We have a manufacturing technical operations team which creates process batch records under the scaled conditions, and a manufacturing team which focuses on operating in a cGMP environment to manufacture the product at high quality standards. So we see these major steps in a successful technical transfer. So it's in order to really increase the likelihood of success, like as I mentioned, effective knowledge transfer of information between the process donor and ourselves is important, and we do this with using technical forms such as questionnaires, technology transfer spreadsheets. And as I mentioned earlier in the previous slide, we perform a facility fit and gap risk assessment. And after these two activities are performed in process, a decision point is discussed between the teams if process changes are needed or additional PD work is required to support these process changes. And frequently, from our experience, like I mentioned earlier, processes are not a plug and play. In my experience, though, it would be best case. But again, frequently, seed train and production bioreactor steps changes are needed and therefore studied in process development prior to entering manufacturing. And these are utilizing established scale-down process models verify sending these processes and confirm effectiveness of process changes. And during these tech transfer programs, we require process flow diagrams, bill of materials, batch production records, media buffer formulation records, and in-process and release methods and specifications. So I'd like to share with you a case study of a recent upstream technical transfer of a seed train and production bioreactor process. And as I mentioned, in the real world, most cases there are some process changes or optimization required prior to entering the suite. In this case study, we'll be sharing with you a monoclonal antibody process using a chill-based fed batch production. So the client's original process utilized a seed train and shake flask and rocking bioreactors, both in the expansion seed train and N-1 N-1 step involving a rocking bioreactor. The production bioreactor of the origin site utilized a 200-liter single-use stir tank bioreactor from a major supplier. The goal of Avid was to transfer the inoculum process to disposable spinner flask while keeping key process parameters consistent, such as expansion triggers and seeding densities. So the Avid process executed disposable spinner flask, followed by an N-1 step of a scaled-up spinner flask, and ending in a 200-liter Sartorius stir tank bioreactor, as I mentioned earlier, is one of our production bioreactors in our manufacturing suite. So as I mentioned earlier, in most processes, this requires some modification or refinement prior to entering manufacturing. So therefore, some studies are required in PD to get the process ready for manufacturing. So in these graphs shown here is the inoculum expansion process as executed in process development. As showing in the left graph is the viable cell count, showing that the process was executed meeting seed train triggers and performance criteria, both in viable cell count trends as well as percent viability. You can actually see on the graph on the right where an improvement in the inoculum expansion process parameters yielded in a improved viability trend on the second iteration of studies. Okay. So when comparing the PD scale down against the historic process manufacturing process data, you can see here that again, with the good preparation on the tech transfer and understanding the process up front, we were able to plan correctly in really executing and replicating, in most cases actually outperforming some of the key process performance indicators such as viability, BCC, and as well as titer. Okay. Now, scaling up from PD to manufacturing. In some cases, in some cases you have to consider again process-dependent process parameters and process-independent scale-independent process parameters to scale bioreactors, such as gassing and aeration, power per unit volume, feed strategies, and so on and so forth. So again, rapid and successful first-time scale-up to manufacturing really is our core competence, and is shown in these graphs as a recent case study showing reproduced viable trends, viable cell count trends, viability, and titer. Okay. As a summary, we are an experienced CDMO providing expertise to evaluate critical parameters in PD as well as to scale manufacturing processes. We involve cross-functional teams in the entire tech transfer process. It is coordinating critical key activities, and it involves detailed discussions between the client and us, involving face-to-face meetings and milestone discussions. And as we shared here as a case study, we were able to execute a comparable process, a seed chain process, as well as a production process, and as a successful tech transfer. That concludes my presentation. Thank you, everyone, for joining the webinar.
Okay, great. Thanks, Roy. So on one of the previous slides, you had a graph, and one of the audience members wants to know what happened to the viability during the seed chain for the blue trend, and what improvements were implemented there.
Yes, sure. So as a key consideration in process transfer is understanding as much of the donor's equipment as well as our equipment we're transferring to. So in most cases, as mentioned, tweaks in the process is required to optimize process performance, such as aeration and gassing strategy. These modifications in the second iteration of experiments enabled us to improve process performance and improve viability and growth. Yep.
Okay. And can you share the types of disposable spinners you use as a reference in seed train and at N-1?
Sure. Yeah. So we are a focused on really transferring to single-use components. So we employ shake flask, disposable agitated spinners, as well as throughout the inoculum expansion train, utilizing stir tank STRs, Eppendorf BioBlues, yeah.
Okay. And what is the typical duration from tech transfer to first at-scale manufacturing batch?
That's a good question. So, as I mentioned earlier, it really depends on a case-by-case basis, where during the technology transfer process, it really is important to point out where gaps and risk is inherent. So if there are modifications to the process, and PD and additional PD studies are required to mitigate the risk of any issues happening in manufacturing due to lack of data. It can require additional time in the timeline. So that is generally discussed in the beginning of the tech transfer program and agreed upon so that a realistic timeline is agreed upon between us and the sponsors. So, you know, in most cases, 4 to 6 months is what I've seen as a duration. Again, it can be longer depending on the phase or the data that is coming from the sponsor side. So, and again, it really is. You know, we're all in the same team to ensure that we have a successful first run. So all that is all the risk is qualified and identified up front, so that we can strategically get it right the first time.
Okay. So we have time for just one more question. If we didn't get to your question, they'll be passed to Roy individually, so you can go ahead and continue to type questions, but this is the last one he will answer. So the last question is Avid strictly clinical producer?
Actually, we are both a clinical manufacturing facility as well as a commercial plant. So we've been producing a commercial product 14 years here at Avid. So we, like I mentioned earlier, we have a very successful regulatory track record, inspected by the FDA, EMEA, over the many years here. So so we have that proven track record, both clinical and commercial.
Okay. Thank you so much, Roy.
Great. Thank you, everyone, for your time and listening in on the webcast.
The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive an email with a follow-up link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.